For the longest time, the war on cancer was fought with pretty much three main weapons. You had surgery, radiation, and chemotherapy. But in the last decade or so, a fourth, and frankly, revolutionary weapon was unlocked: immunotherapy. And this was a totally different approach. It wasn't about poisoning the disease. It was about unleashing our own immune systems to kill cancer. And the results? Well, they completely changed the game.
I mean, just look at metastatic melanoma. Not that long ago, a diagnosis like that was basically a death sentence. But today, for patients on a combination of immunotherapy drugs, the 5-year survival rate is hovering right around 60%. Just let that sink in. These are people who were told they were out of options, and now they're watching their tumors literally vanish, living full lives years later. It's an absolutely stunning victory.
But that incredible victory brings us to a huge mystery. For all of its power, immunotherapy only seems to work for a minority of patients, and only for certain kinds of cancer. So the biggest, most urgent question in cancer research right now is, why? Why do some people have these, miraculous responses, while so many others don't respond at all?
To get to the bottom of that, we have to look way closer. We need to completely throw out our old image of what a tumor is. It is not just a simple blob of rogue cells. Think of it more like a dynamic, complex, and really hostile ecosystem.
Scientists have a name for this: the tumor microenvironment, or TME for short. Now here's why this is so important. It reframes the entire problem. The cancer cells, yeah, they're the enemy. But the TME is the fortress they've built to protect themselves. And it's a fortress complete with defensive walls, supply lines, and even local cells that have been corrupted and turned into traitors.
And get this, we're not talking about some minor detail here. In some of the toughest cancers to treat, like pancreatic cancer, up to 90% of the tumor's mass isn't even cancer cells. It's all this other stuff, this stroma, this whole neighborhood of corrupted cells and structures that are there just to support the cancer.
And that battlefield is seriously crowded. You've got the cancer cells, obviously. You've got our immune cells, like the T cells, who are trying to get in there and do their job. Then you've got these stromal cells, like fibroblasts, the blood vessels that act as supply lines, and the whole thing is held together by this physical scaffold called the extracellular matrix. It's a real mess.
So when scientists first started digging into this puzzle, their first idea was pretty straightforward. They focused on the most obvious interaction, the one between the cancer cells and the immune cells.
The thinking went something like this. Our immune T cells are the soldiers. The more mutations a cancer cell has, the more foreign it looks, and the more little red flags it raises that the T cells can spot. So, immunotherapy drugs, these things called checkpoint inhibitors, just cut the brakes on the T cells, letting them go out and destroy anything that has a red flag. It's simple, it's elegant.
And what's so cool is how the technology we were developing ran right alongside these discoveries. As our ability to sequence DNA got faster and cheaper, especially after the Human Genome Project, we could finally start counting up all those genetic red flags and connect the dots between how many mutations a tumor had and whether a patient would respond to the treatment.
And the data, as you can see right here, was really convincing. It's a clear correlation. Tumors with a low number of mutations, they had a low response rate. Tumors with a really high mutational burden, like melanoma, had the best response rates. The more flags for the T cells to see, the better the drugs worked. It all seemed to make perfect sense.
Except that wasn't the whole story. Not even close. Because researchers kept finding these weird cases. Tumors that were packed with mutations, with tons of T cells right nearby, but the treatment would just completely fail. The army was right there at the fortress gates, armed and ready to fight, but some kind of invisible force was keeping them from getting in. So the number of flags wasn't the only thing that mattered.
And this is where the investigation took a really fascinating turn. Instead of looking at the soldiers or the enemy, scientists started examining the fortress itself. And what they found was a hidden accomplice, a bodyguard, actively protecting the cancer cells from the inside.
When you actually look at tumor biopsies, you see three distinct patterns. Some tumors are what we call immune deserts. The T cell army is just nowhere to be found. Others are inflamed, which is good. The T cells are inside, mixed in, fighting hand to hand. But the key to our mystery is this third type, the excluded tumor. The T cells are there, they've made it to the battle, but they are physically trapped outside the tumor, held back by some kind of defensive wall.
So who's building this wall? Well, meet the cancer-associated fibroblasts, or CAFs for short. These cells actually start out as normal cells that are supposed to help build and repair our tissues. But the tumor hijacks them with chemical signals, turning them into enemy engineers.
And instead of helping the body, they start building this dense, physical fortress to protect the cancer. And this is literally how they do it. First, the tumor sends out a signal called TGF-beta. That signal activates the fibroblasts, telling them to multiply and start pumping out huge amounts of this thick, web-like material. This material forms a literal, physical barrier, a dense wall that our T-cell soldiers just cannot push through.
So this leads to a very logical next question, right? If these CAFs are the enemy engineers building the wall, why not just take them out? Just develop a drug to destroy them, and the problem's solved.
So they did exactly that. In mouse models of cancer, they figured out a way to completely wipe out these CAFs. The hope, of course, was that the fortress would just crumble and the tumors would finally be vulnerable.
But the result was the absolute opposite of what anyone predicted. When they destroyed the calves, the cancers became more aggressive. The mice died faster. It was this stunning, unbelievable twist. Getting rid of the engineers somehow made the fortress even deadlier.
This shocking result forced this profound shift in thinking, and it's captured perfectly in this quote from a leading researcher, Dr. Elizabeth Jaffe. The goal isn't brute force. It's not about killing the fibroblasts. The goal is to be smarter. You have to reprogram them, turn them from enemies back into allies. And the key to reprogramming them is understanding that not all CAFs are the same. It turns out there are different subtypes. You have MyCAFs, over on the left here, and they're the main wall builders. But you also have iCAFs, which are more like propaganda officers, sending out signals to suppress the immune system in other ways. They have different jobs, which means we can target them in different ways.
So this much deeper, much more nuanced understanding of the tumor's fortress and its cellular bodyguards, well, it's now paving the way for a whole new generation of much, much smarter cancer therapies.
We are really entering a new era. The strategy is moving beyond just boosting the T-cell army. Now it's a more sophisticated two-pronged attack. Energize the soldiers and, at the very same time, systematically start dismantling the enemy's defenses. And this is what that new playbook looks like in the real world. You can see trials are already happening that combine standard immunotherapy with drugs that block that TGF-beta signal, stopping the wall from ever being built in the first place. The ultimate goal here is total personalized medicine. To one day be able to analyze a patient's tumor, see exactly what kind of fortress their cancer has built, and give them a custom cocktail of drugs designed to tear it all down.
So the reason immunotherapy so often fails isn't just about the soldiers or the cancer cells, it's about the battlefield itself. And by learning how to reshape that battlefield, to literally turn cancer's own bodyguards against it, we are unlocking an entirely new and powerful front in the war on cancer. Which leaves you with one last really fascinating thought. If we've just figured this out, what other secrets is that fortress still hiding?